selection, where the environment changes, where instead of being just an ordinary background where they live, the background has turned darker, and so these mice can blend in with the environment better, hide from predators. The lighter ones stick out, and so they get eaten, so they get removed, so natural selection shifts. In diversifying, the middle gets removed. So the environment, they have adapted to two different environments, a light and a dark one. The mid one is pulled out, and then stabilizing is when the extremes are selected against, and everything shifts to a single phenotype.
Just narrow them down.
Narrows it down and stabilizing. So those are three different ways in which natural selection can occur. Okay. Where am I at? Okay. All right.
Turtle.
Yeah. This is going to go fast. The last part here. That was okay. Okay. Now we're going to look at how species came about. Two different types of speciation. We have anagenesis and cladogenesis. In this one, in anagenesis, you just have an accumulation of traits, and it's like a single change from one species over time just to a single one. One gives rise to another in anagenesis. That's all it is. It's just a buildup of changes. And then versus cladogenesis, we're going to build a cladogram a little bit later, but what happens is one species serves as a common ancestor to two distinct ones later on, where this one survives in addition to a new form. In this one, the original form dies and the new form is able to survive. That's kind of the difference between the two. Clado pretty much means to pretty much diverge, to branch, and then the ana is just the single. I already talked about the biological species concept. Anything that can reproduce will be able to, and that's a species.
Yeah.
Okay, you can read that over.
Yeah.
I put this in. You don't even need to know this one. You can just skip those. I just put those in. Okay, modes of speciation. This was a very distinct thing. One of the articles was all about that. Allopatric versus sympatric speciation. In allopatric, some type of natural condition occurs to separate a population, such as a big canyon in between those two populations of trees. The environment changes on each side, and so each one adapts differently, and so which causes a change. So any type of geographical barrier causes allopatric. Is geographic. Sympatric is a genetic change within a population. Some organisms within a population mutate, change, and then they just develop within a single population. Question?
About the slide with birds, the claudiogenesis, you were saying the other one's single and...
Yeah, the anagenesis is, oops, go back. Yeah. So what happens in anagenesis, you have a single ancestral form here, and what happened is over time they've changed. This original one has become extinct, no longer adapted. All that's left is the new form. Versus cladogenesis, where the original form still survives and produces another form, and it branches off down here. So both have been able to adapt, versus only one has been able to adapt.
Okay.
Then there you go. Another one is punctuated equilibria, kind of how things change. There's gradualism versus punctuated. Gradualism is pretty much just a gradual change over time. That's why it's called gradualism. You see different intermediate forms in the organism until they branch and diverge that way, versus the punctuated, where there's massive changes in phenotype and characteristics over time. And kind of going back in the reading, it was Gould and Eldridge kind of used that, figured out kind of how that occurs, and where you have usually a lot of environmental, ecological disturbances causes those major changes versus the gradual. So it's usually an ecological cause for that. Okay, now we'll hit Evo-Devo real quick, evolutionary development. pretty much what this is developmental genes are similar amongst a lot of different organisms. And that's why when I showed you the pictures of the embryos that's why humans are born with tails sometimes, is because we all have similar genes. That's evolutionary development. Okay. And so one type of growth is called allometric growth, and this is evo-devo, in which proportions of body size change over time. So when you look at the baby, a quarter of its body length is a head. And then as it moves down, the head becomes smaller and smaller. That's allometric growth. A lot of organisms do that. And that's similar genes are at action to change that. Versus, here's another one. Another allometric pattern, you're looking at the chimp fetus and the human fetus. You know how all the bones in the head are kind of not fused together. And then over time, though, this all happens to help get to the birth canal. And then over time, the head fuses together in the skull and it kind of changes. So you can kind of see the human fetus and the chimp fetus, very, very similar. Not much difference. And then so those are similar genes in action.
Changes a lot, the chimpanzee.
Yeah, the chimp
Change
changes.
that other shape.
Exactly, yeah. And so that's where part of our 1.5% difference between us and the chimp comes in, is that change in the adult skull. But the only way to get this is similar genes, which causes a common ancestor. Another one is called a heterochrony. And this is timing of developmental events. What happens is you have two different, like salamanders, two different ways. You have ground dwelling and a tree dwelling. The tree dwelling one have more of the webbing, like some humans are born with webs on their fingers. Same thing here, they are able to survive better, the tree dwelling ones with the webbed versus the ground dwelling one, which is a little less webbed in there. So that's heterochrony. Yeah, don't worry about that. Okay. Another one is something called the homeotic genes. And what these do, it's also known as a Hox gene. You'll see Hox probably pop up in the super quiz. It's called, which stands for homeobox, which is a set of genes that code for turning on a gene. So homeobox genes turn genes on, they regulate them, and so they just kind of abbreviate it as Hox. And what these do, it tells the organism where to put certain appendages, like legs, arms, antennae, wings, and each of these Hox genes changes. You can actually pull a Hox gene like out of fruit flies. You can pull the wing gene out, put it on its head in development, and actually grow a wing out of its head by changing the Hox gene. You can change the legs around, put legs on its head, put it all these different areas. Okay, and so those are what the Hox do. And so, let's see here. What did I do on this one? So yeah, these are just other ones where the different Hox genes, so you can kind of see where the different region is, and then wherever they're located, it changes the expression where they want to grow those certain genes, like the fish fin versus the fingers or the legs of the chicken versus the fish. So it determines development. Okay, so that's Hox. What else do I got? Yeah, don't worry about that, you can read that. Okay. Almost done. Couple more. Okay, so far so good. I know I'm flying through this, a lot of information. Any questions so far? Doing okay?
Yeah.
Okay. All right, this is a small part. Oh, yeah. Phylogeny and systematics. Kind of how this is the evolutionary history of species, for the most part. Systematics is just looking at evolutionary history in context, kind of how things are related and when they were related. And a lot of this uses the fossil record in order to figure this out, different strata of the fossil record to figure out where they're located. So we're looking at phylogeny here. So when looking at the fossil record, just a quick background, it's usually in sedimentary rock. Fossils found on lower levels, lower sedimentary levels are older than the ones on top.